morning, everyone. This is Gia's Academy. Let us see the mechanism of sorting and the regulation of intracellular transport from Unit 2. Let us see the first one. What is one large sorting system? One large sorting system means the protein biosynthetic pathways in cells can be considered one large sorting system. Okay, that is what you are seeing here is the endoplasmic reticulum. Endoplasmic reticulum is releasing the vesicle which is taken to the Golgi apparatus, and from the Golgi apparatus the proteins are released outside by the secretory vesicles, and this secretory vesicles are come in contact with the plasma membrane, and these proteins are released outside. Okay, this is the path of the protein. So this is called as one large sorting system. The protein sortage is called as, okay, sorting is called as one large sorting system. What is pre-protein? Any protein that contains a targeting sequence that is subsequently removed is designated as the pre-protein. And number 3, what is secretory or exocytotic pathway? Okay, what is exocytotic pathway? If the question asks like that, or what is secretory pathway, the question comes like that, then your answer is the entire pathway of endoplasmic reticulum, from endoplasmic reticulum to the Golgi complex, okay, and from the Golgi complex to the plasma membrane, that is endoplasmic reticulum to Golgi complex and Golgi apparatus to the plasma membrane is often called the secretory or exocytotic pathway. Okay, exocytotic in the sense given out from the cytoplasm or cytosol. Okay, so it is called as exo. Exo means out. Cytotic in the sense from the cytoplasm to the plasma membrane. It is called as exocytotic or secretory pathway. Okay, it is secreted outside through the Golgi apparatus. Golgi apparatus is producing the secretory vesicle and it is given out. The number 4 is what is constitutive. What is constitutive? That is the secretory pathway involving transport vesicle is called constitutive. Okay, transport vesicle, this path is called as constitutive. Okay, this part is called as constitutive, and this part is called as secretory pathway. Okay, this is secretory pathway, and this is called as constitutive. Okay, this is called as constitutive. So the entire pathway from the endoplasmic reticulum, Golgi apparatus, plasma is called the secretory or exocytotic pathway. Okay, and these are called as transport vesicle and these are called as secretory vesicle. Okay, now translocation. What is translocation? Let us see. Translocation is passage through the two membrane. Okay, passage through the two membrane is called translocation. Which one? The protein. Okay, protein. If you take, this is the mitochondria, this is the outer membrane of the mitochondria, and this is the inner membrane of the mitochondria. This is interspace between the two membrane, and this is the mitochondrial matrix. Okay, mitochondrial matrix. What you are seeing here is the mitochondrial matrix. This is cytosol, okay, or the cytoplasm. Now the proteins from the cytoplasm is entering into the mitochondria through the outer membrane, and there is a special structure what it is called as TOM, and then it is entering into the next structure that is called as TEM. Okay, TEM. So there is a passage through that the protein from the cytosol is entering into the mitochondria through the TOM and TEM. Okay, and it is coming to the mitochondrial matrix. Okay, so how many proteins? In the sense, there are 13 proteins. Okay, there are 13 proteins. These proteins are mostly membrane component of electron transport chain. Okay, these are present on the mitochondria, and these proteins are encoded by the mitochondrial genome and synthesized in that organelle using its protein synthesizing system. Okay, most progress has been made in the study of protein present in the mitochondrial matrix. Okay, most of the protein present in the mitochondrial matrix, such as in the F1 ATPase subunits. You can see a subunits like this, ATPase subunits. Okay, most of the proteins are present in the mitochondrial matrix, such as the F1 ATPase subunits. Okay, so translocation in the sense, the protein from the cytosol which is coming inside through the mitochondrial membrane, outer membrane, as well as the inner membrane, that is called as translocation. Okay, and TOM means translocase of the outer membrane, translocase of the outer membrane. TIM in the sense, translocation of the inner membrane. Now, what is Zellweger syndrome? What is Zellweger syndrome? See, what you are seeing, this child is affected by that syndrome. This is a condition inapparent at the birth, and the child die within a year. Okay, how this syndrome is occur? In the sense, this syndrome is also known as cerebrohepatorenal syndrome. That is, it affects the brain, it affects the liver, and it affects the renal system also. Okay, so it is called as cerebrohepatorenal syndrome. Okay, this name is very important. Why? In the sense, what is the other name of Zellweger syndrome? In the sense, the other name is cerebrohepatorenal syndrome. Okay, what are the organs affected by this syndrome? In the sense, the brain. Okay, the first organ is the brain, second organ is liver, and the third organ is kidney. Okay, kidneys. So these are the organs. These three organs affected, in the sense, naturally the life will be ended. Okay, so this syndrome is otherwise called as cerebrohepatorenal syndrome. So what are the organs, main organs it affects? In the sense, these three organs will be affected. So the child within a year, its life comes to an end. Okay, and this is an inherited disorder. Okay, that also make a note, it is an inherited disorder. Okay, the genes will be, if there is any mutation in the genes, then this occur. And also one more thing you have to think, the absence or reduction of functional peroxisomes. Okay, the major reason for this syndrome is absence or reduction of functional peroxisomes. Okay, what is peroxisome? In the sense, peroxisome is a cell organelle. Okay, cell organelle, what you can see here. See, this is the peroxisome. If you are taking this cell, and you can see the peroxisome here, it is one of the cell organelle. Okay, one of the cell organelle. So if there is any reduction or absence of function of peroxisomes, leads this syndrome. Okay, so what is the syndrome called? Zellweger syndrome. It is also known as cerebrohepatorenal syndrome. Okay, this is mainly because of the absence or reduction of peroxisomes, functions of peroxisomes. Okay, peroxisomes are a cell organelle which is present in the cytoplasm of the cell. Okay, and come to here, biochemical findings include an accumulation of very long chain fatty acid. Okay, this is also another reason for this Zellweger syndrome. Okay, accumulation of accumulation of very long chain fatty acid as well as abnormalities of the synthesis of bile acid, okay, are marked the reduction of plasmalogens, okay. So these three you have to keep in mind. First one, the absence or reduction of functional peroxisomes, and the second one is accumulation of very long fatty acid and abnormalities of the synthesis of bile acids, okay. So these are the thing. And if you come to here, the condition believed that due to mutation of the genes, this already I told. So mutation, so number 1, number 2, okay, number 2, and the bile acid is number 3, and this is the fourth reason, that is mutation of genes, okay, for certain proteins called peroxins, okay. And two closely related conditions are neonatal adrenoleukodystrophy, okay. So and infantile refsum disease, okay, these also very closely related to this syndrome. These two diseases are also closely related to this syndrome. Which one? Neonatal adrenoleukodystrophy and infantile refsum diseases, okay, these two are also very closely related to this syndrome. Now let us see what is peroxisomes, okay. So it's an organelle I told, and some disorders occur in this peroxisomes leads to that syndrome, okay. Now this peroxisome contains 15 different enzymes. Noted how many enzymes? In the sense 50 different enzymes are present in peroxisome, okay, which are involved in variety of biochemical pathways in different types of cells. And usually it synthesizes the hydrogen peroxide, and this hydrogen peroxide, if it is synthesized in a large amount, it is a toxic substance, so it has to be decomposes, okay, it has to be decomposes. So what happened, it convert, this peroxisomes convert this hydrogen peroxide into water, or by using it to oxidize into another organic compound, okay. So immediately what this peroxisomes does, in the sense hydrogen peroxide is also important, but if it is in large amount it is very toxic, so immediately it convert that into water or another organic compound, okay. And in animal cells, if you see, the fatty acids are oxidized in peroxisomes as well as in mitochondria. So fatty acids oxidation also takes place in the peroxisomes, okay. You can see this one clearly, the peroxisomes. See, sometimes these peroxisomes are also affected, okay. How? In the sense, it is, if it is a single enzyme or protein deficiency, in the sense this will affect only a certain part of the peroxisome, okay. But the peroxisome biogenesis disorder, in the sense this affect the overall assembly and function of the entire peroxisome. At that time what happened, peroxisome parts, all the parts are affected, and it is not performing properly, and all the parts cannot do their jobs, so that syndrome occurs, okay. So if it is a single enzyme, in the sense some part will be, or if it is completely affected, in the sense it leads to that disorder. What is that? That syndrome, okay. What is that syndrome called? It is called as Zellweger syndrome, okay. Zellweger syndrome, it is otherwise called as cerebro-hepatorenal syndrome. This is also associated with two other diseases, that is neonatal adrenoleukodystrophy and infantile refsum disease, okay. These two are also accompanied with this one, okay. Coming to the signal hypothesis explains how polyribosomes bind to the endoplasmic reticulum. Okay, the signal hypothesis was proposed by Plobel and Sabatny. Okay, this one noted. Blobel and Sabatini. Okay, Sabatini. Okay, which one? The signal hypothesis. Signal hypothesis was proposed by Global and Sabatini. Okay. Now, protein synthesized on membrane-bound polyribosome contained a peptide extension. Okay, peptide extension and a signal peptide. Okay, so what is happening here? The protein synthesis is a long chain, and when it come to the abstraction, the protein synthesis will stop. Okay, and what is elongation arrest in the sense? See, this is an long protein, so this protein synthesized is going on, and if it is receive, or if it is receive any signal from the signal recognition particle, okay, that blocks the further translation. About 70 amino acids have been polymerized. Okay, this block is referred as elongation arrest. Okay, the blockage is referred as elongation arrest. Okay, and now let us see what is co-translational insertion. Co-translational insertion. Okay, the signal hypothesis proposed that the protein is inserted into the endoplasmic reticulum membrane. At the same time, its mRNA is being translated on polyribosomes, so it is called as co-translational insertion. Okay, so what is happening here? The protein is inserted into the endoplasmic reticulum membrane. Okay, protein is inserted into the endoplasmic reticulum membrane. Okay, is being translated on polyribosomes. At the same time, its mRNA is being translated on polyribosomes, so it is called as co-translational insertion. Okay, this insertion is called as co-translational insertion. And SRP imposed block is not released until the SRP signal peptide E polyribosome complex has bound to so-called dogging protein, that is called as dogging protein on the ER membrane, endoplasmic reticulum membrane. And this SRP thus guided the signal peptide to the SRPR and prevent the premature folding and expulsion of the protein being synthesized into the cytosol. Okay, so this is translational insertion. Okay, next one, see what is meant by TRAM. What is meant by TRAM? TRAM in the sense translocating chain associated membrane. Okay, translocated chain associated membrane. Okay, so protein may bind the signal sequences. It is initially interacted with the translogon. What you are seeing here is the translogon, and the secretary. What is meant by TRAM? Translocating chain associated membrane, it is called. Okay, TRAM in the sense translocating chain associated membrane. It's a protein. It may bind the signal sequences as it initially interact with the translogon and the ACE61 complex. Okay, consisting 3 proteins which bind the heavy subunit of the ribosome. Okay, the insertion of the signal peptide into the conducting channel. Okay, insertion of signal peptide into the conducting channel while the other end of the protein is still attached to the ribosome. Can you see this? This is the peptide which is attached to the ribosome. The other end is attached to the ribosome. Okay. This is termed as co-translational insertion. This type of insertion is called as co-translational insertion. Okay. The process of elongation of the remaining portion of the protein probably facilitates the passage of the nascent protein across the lipid bilayer. This is endoplasmic reticulum actually. This is the membrane of the endoplasmic reticulum. The elongation of the protein takes place. Probably it is facilitated the passage of the nascent protein across the lipid bilayer as the ribosomes remain attached to the membrane of the endoplasmic reticulum. See, this ribosome is attached to the membrane of the endoplasmic reticulum, and this protein elongation is taking place here. Okay. So tram means translocating chain associated membrane. So this membrane is called as translocating chain associated membrane, and this is that protein. Okay, it is called as translogon. Translogon. Okay. And what is happening inside here in the sense insertion of signal peptide into the conducting channel. This is the conducting channel, while the other end of the parent protein is still attached with the ribosome. So this is termed as co-translational insertion. Okay. And you can see the various translocation takes place in various parts of the organelle. Okay, very different organelle. This is ER translocation. Okay, this is endoplasmic reticulum. Here the translocation of protein occurs. Okay, and if you come here, this is chloroplast import. That is the protein from outside in the sense in the cytoplasm, enter into the chloroplast, and it is moving towards the specific sites of the chloroplast. Okay, and here it is mitochondrial import. See, from the cytosol, the chaperone is taking the protein, that is mitochondrial precursor protein, it is taking and it is inserting here. This is the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. This is the tom and this is stem. So it is coming through this. It is the inserted inside the membrane. Okay, through the translocase of outer membrane through the TIM, and then it is coming to the, this is tom and this is TIM. Okay, inner membrane. So it is coming here and entering into the matrix. Okay. So this is the import chaperone and this is cytosolic chaperone. Okay, it is receiving and it is bringing to the particular tom. Okay, so these are the various organelle. How this translocation takes place is given nicely here. Okay, and if you come here, this is a general model. The chaperone is receiving the or bringing the protein to the receptor. Okay, to the receptor. So this is again the signal peptide which is coming and attached to this translogon. Okay. And what is LDL receptor? Okay, what is LDL receptor in the sense? The LDL receptors enters the endoplasmic reticulum membrane in a manner of, okay, analogous to a secretory protein. It is just like the secretory protein. It partially transfers the endoplasmic reticulum membrane and it signals the peptide which is cleaved and its amino terminal protrudes to the lumen of the endoplasmic reticulum. Okay. See, it is these are the receptors, LDL receptors, which is receiving the protein, and just like endocytosis, it is entering inside. Okay, and then it is reaching the endoplasmic reticulum. These are the amino acids. It is taking the amino acids and reaching the endoplasmic reticulum. So this receptor is called as LDL receptor. Okay, LDL receptor. So thanks for everyone, and go through each and every part of this class, and the part 1 and part 2 are given in the description box, as well as in our app it is uploaded, so you can verify that and you can prepare for the examination. Okay, all the best.